Welcome back. In this session, I will be discussing about epithelial glands. What are epithelial glands? What are the functions of epithelial glands? Based on what features are they classified? And I hope you will be curious to know the general structure of the glands. The differences between various types of epithelial glands, and are there any abnormalities of these glands? If so, what are they? All these questions will be answered in this series of sessions on the epithelial glands. The learning outcomes of this series of sessions are, by the end of the session, you should be able to define the epithelial glands, mention the different types of epithelial glands based on the number of cells, shape of secretory element, mode of secretion, nature of secretion, and presence or absence of ducts. Under the general structure, explain the connective tissue framework, parenchyma, and duct system of the glands. Along with the details, you should know the functions of epithelial glands and understand the applied anatomy of epithelial glands. To start with, let us see what are epithelial glands. Epithelial glands are organized collections of secretory cells specialized to perform a secretory function. Such cells are present in single or in groups. The secretory products are stored in membrane-bound vesicles called secretory vesicles. What are the functions of epithelial glands? The secretory cells may synthesize, store, and release proteins, which is like in the pancreas, lipids, which is seen in the adrenal and sebaceous glands, or complexes of carbohydrates and proteins as in salivary glands, and epithelia of mammary glands secrete all three substances. Now let us see the classification of epithelial glands. Glands can be classified broadly into endocrine and exocrine glands. Let us first discuss about the endocrine glands. These are ductless glands and secrete various hormones and other useful chemical substances into the blood capillaries. So the secretions are poured into the capillary plexus surrounding them. The gland consists of epithelial cells supported by reticular fibers and permeated by rich fenestrated capillary plexus. The glands may be of two types, cord or clump type and the follicular type. In the cord or clump type, the cells are arranged in irregular cords and clump and store the secretion inside the cell by intracellular method. From inside the cell, the secretion is directly delivered outwards into the capillaries. Most of the endocrine glands belong to this type. In the second type of endocrine glands, which is follicular type, the cells are arranged in single layer on basement membrane surrounding the central lumen. The cells are called follicular cells and all these together is called as follicle. Luminal surface of the follicular cells are provided with microvilli, and outside the basement membrane, the follicles are surrounded by intense capillary plexus. The cells secrete and pour the secretion inward into the lumen and form a pool of stored secretion by extracellular method. So the secretion is stored in the center of the follicle surrounded by cells. This type of follicles are present in thyroid gland and is called thyroid follicles. Now let us discuss about exocrine glands. Glands that pour their secretions onto an epithelial surface directly or through ducts, are called exocrine glands. Based on the number of cells, glands may be classified into unicellular or multicellular glands. Before going into further details of classification, let us know how the glands develop. Both exocrine and endocrine glands develop as diverticula of the epithelium. The exocrine gland develops as a solid bud from the epithelium into the underlying connective tissue. Soon it elongates, undergoes canalization by disintegration of the cells in the center of the cord, and the remaining cells at the periphery differentiates into secretory and conducting portion. The cells at the lower part of the cord differentiates itself into secretory cells, and the cells in the upper part or the conducting part close to the surface differentiates into duct cells and forms the duct and is continuous with the epithelium. Hence, the exocrine gland discharges its secretion through a duct. Endocrine gland also develops in a similar manner to exocrine gland up to proliferation of solid cord of cells invaginating into connective tissue. The solid cord of cells which was to develop into duct disappears and breaks the continuity with overlying epithelium. The cells in the lower part develop into two types of endocrine glands. First, the cells arrange irregularly and appears as a clump. Soon these clumps of cells get surrounded by blood vessels into which they pour their secretions. This type of cells in the form of clumps or cords are present in islets of Langerhans in pancreas, suprarenal gland, pituitary gland. Okay. In thyroid gland, the cells in the center of the clump disappears, okay, forming the lumen. Only a single layer of cells rest on basement membrane surrounding the lumen in the center, forming secretory acini. These acini are surrounded by the blood capillaries. Now, coming back to classification of glands, the exocrine glands are further subclassified based on the number of cells into unicellular and multicellular, as we have already seen. Unicellular glands are interspersed as goblet cells amongst other non-secretory epithelial cells, okay, such as simple cuboidal, simple columnar and pseudostratified epithelia. The goblet cell rests on the basement membrane. It has an elongated nucleus at the base. The base is narrow and the apical portion is broad due to accumulation of secretory granules. They secrete mucus and are found in the epithelium lining the intestines and respiratory tract. The multicellular glands are groups of cells resting on basement membrane with lumen in the center, having same structure and secretory functions. Most glands are multicellular in structure. It has two portions. The cells in the lower part of the gland is the secretory cells surrounding the lumen, and the part close to the surface is ductal cells surrounding the duct. These type of glands are present in lacrimal gland, parotid gland and many other glands. Based on the branching of ducts, the multicellular glands are classified into simple and compound glands. When all the secretory cells of an exocrine gland discharge the secretion into one common duct, the gland is said to be simple gland. These type of glands are present in gastric glands and sweat glands. In compound glands, there are a number of groups of secretory cells, each group discharging into its own duct. These ducts unite to form larger duct that ultimately drain onto an epithelial surface through a single duct. These types of glands are present in parotid gland and pancreas. Based on the shape of the secretory units or portions, both in simple and in compound glands, the secretory cells may be arranged in various ways and are classified into tubular, acinar or alveolar and tubulo-acinar or tubulo-alveolar glands. In tubular glands, the secretory unit is in the form of tubule. The tube may be straight or coiled. So these type of tubular glands are present in the gastric glands. In acinar or alveolar glands, the secretory units are in the form of blind dilated ends with round or oval or flask in shape. They are present in the salivary glands. In tubulo-alveolar glands, there is a combination of both types having tubular and alveolar secretory portions. Okay, these glands are present in parotid and submandibular glands. Based on the variations of the ducts and the secretory portions, it is classified into simple tubular, simple acinar or alveolar, compound tubular, compound acinar or alveolar, and compound tubulo-acinar or tubulo-alveolar glands. Simple tubular gland has one duct with secretory portion in the form of tube. Simple acinar or alveolar gland has one duct with secretory portion in the form of acini or alveoli. Compound tubular gland has numerous secretory portions in the form of tubes having their own ducts. Compound acinar or alveolar gland has numerous secretory portions in the form of acini or alveoli having their own ducts. Compound tubulo-acinar or tubulo-alveolar or racemose gland has both tubular and acinar secretory portions having their own ducts. Based on the nature of secretions, it is classified into mucus and serous gland. In the mucus glands, most of the acini are of mucus type. Cells of mucus acini are tall columnar in shape. Flat nuclei are present close to the basement membrane. The lumen of the mucus acini is larger than the serous acini. In mucus glands, the secretion contains mucopolysaccharides in the form of mucinogen granules which collects in the apical parts of the cells. As a result, nuclei are pushed to the base of cell and are compressed against the basement membrane, hence appears flattened. In histology, slides stained with hematoxylin and eosin stain, the secretion within the mucus cells remains unstained or pale stained, so that cytoplasm is colorless after staining and they have an empty look appearance. This is because during the preparation of the slides, the mucinogen granules are washed off. In serous glands, the acini are of serous type. Cells of serous acini are triangular or pyramidal in shape with a rounded or spherical nucleus placed in the lower half of the cell. Zymogen granules are present at the apex of cell. The lumen of the acini is small. The secretions of serous glands are protein in nature. With hematoxylin and eosin stain, the cytoplasm of these cells often stains bluish and called basophilic cytoplasm. Apex stains bright pink, hence called eosinophilic granules. Based on the mechanism of releasing the product, it is classified into merocrine, apocrine and holocrine glands. In merocrine or ecrine or epicrine glands, okay, the secretions are thrown out of the cells by a process of exocytosis with the cells remaining intact. The secretions are wrapped in the membrane-bound vesicles called secretory vesicles, which moves towards the apex, okay, and coalesce or merge with the cell membrane and release the product, okay, outside the cell. So these type of glands are seen in the goblet cells. Okay, the goblet cells release the secretion in the form of exocytosis. Okay, so they come under the merocrine glands. Okay, the other example for merocrine gland is the salivary gland. The second classification based on the mechanism of releasing the product is apocrine gland. In these glands, the apical part or luminal part of the cells are pinched off from the cell with secretory particles disintegrating during the process of secretion and are shed off to discharge the secretion, leaving behind the nucleus and basal part of cell from which the apical disintegrated part of cell regenerates. So these type of glands are present in sweat gland and mammary gland. The third classification based on the mechanism of releasing the product is holocrine gland. In these glands, the entire cell or the whole cell along with secretory particles undergo apoptosis or self-destruction or disintegrates and die while discharging and become part of secretion itself. Sebaceous gland is a good example for holocrine gland. And coming to the last classification of multicellular exocrine glands, which is based on the development, that is which germ layer it develops from. It is classified into ectodermal, mesodermal and endodermal glands. If the gland develops from the germ layer ectoderm, it is called ectodermal gland. Glands of the skin, mammary gland, lacrimal gland and salivary glands along with hypophysis cerebri, chromaffin organs develop from ectoderm. If the gland develops from the germ layer mesoderm, it is called mesodermal gland. Suprarenal cortex, gonads, kidneys, spleen develop from the mesoderm. If the gland develops from the germ layer endoderm, it is called endodermal gland. Thyroid, parathyroid, thymus, liver, pancreas, lining cells of all glands of the alimentary tract excepting salivary glands, respiratory tract, prostate, urethral, bulbourethral and greater vestibular glands develop from endoderm. Till now, we have discussed the definition and classification of epithelial glands based on the presence or absence of ducts as endocrine and exocrine glands, based on number of cells as unicellular and multicellular glands, and subclassification of multicellular exocrine glands based on the shape of the secretory portion as tubular and acinar, based on the branching of ducts as simple and compound, based on the nature of secretion as mucus and serous, based on the mechanism of releasing the product as merocrine, apocrine and holocrine, based on which germ layer it develops as ectodermal, mesodermal and endodermal glands. In the next session, I will be discussing on different types of major salivary glands and its structure. Join my Facebook community, link in the description. Like, share and subscribe. Thank you for watching.